Hey everyone, and welcome to today's Explainer. You know, every single day, your body goes toe-to-toe with countless foreign substances. But here's the thing, your adaptive immune system doesn't just throw up some generic wall of defense. No, it operates with absolute microscopic precision. So today we're diving straight into the architecture of immunity. We're going to demystify specificity, which is the exact, frankly, mind-blowing process your body uses to identify and take down individual microscopic enemies.
I want you to really think about this for a second. Imagine you encounter a never-before-seen virus. How on earth does your immune system know exactly how to target it without accidentally friendly fire in your own healthy cells? If your innate immune system is kind of like a broad security net looking for general trouble, your adaptive immune system is like a master detective hunting for a highly specific fingerprint. How does it manage to tell apart literally millions of completely distinct molecular structures, down to the tiniest variations in a single bacterial strain? Well, that is the foundational puzzle we are solving today.
Here is our roadmap. We'll be looking at the power of recognition, B cells and T cells, the clonal selection theory, the necessity of diversity, cross-reactivity and nuance, and finally, the targeted immune response.
Okay, let's jump right into Section 1, the power of recognition, choosing the target.
So look at it this way. When antigen X enters your body, only the lymphocyte specifically designed to recognize X is going to respond to it. If antigen Y shows up, a completely different Y-specific lymphocyte jumps into action. This selective recognition is exactly what we mean when we talk about specificity. An immune response isn't just a simple question of whether the body is going to react. It's this highly calculated, deliberate process of choosing exactly what to react to. This right here is the absolute starting point of all adaptive immunity.
Moving on to Section 2, B cells and T cells, the hardware of immunity.
So our two main defenders, B cells and T cells, actually operate very differently from one another. While both handle specific immunity, what it literally means for them to see an antigen is completely different. B cells use their B cell receptor, or BCR, to just directly spot specific structures on proteins and molecules floating freely around your body. And when they get activated, they eventually start pumping out antibodies that share that exact same specificity. T cells, though, they're a bit more high maintenance. Their T cell receptor, or TCR, requires the antigen to be chopped up into tiny little pieces, peptides, and physically presented to them on the surface of other cells by molecules called MHC. It's like serving it on a silver platter. But for both of these cells, the golden rule remains. Specificity always starts with these receptors.
Now, your body has millions of these lymphocytes, right? But they do not all have the same receptor. During development, each cell forms a receptor with one highly specific target. A single lymphocyte and all the subsequent cells that multiply from it are going to share that exact same antigen receptor specificity. We call this specialized group a clone. Before an infection even happens, your body already has numerous unique groups of these cells, distinct clones, just waiting in the wings. Conceptually, it's like you've got clone A waiting around for antigen A, clone B waiting for antigen B, and so on.
Which brings us to Section 3, the clonal selection theory, the paradigm shift.
Okay, this is the massive aha moment. When a foreign invader gets into your body, your immune system does not suddenly analyze it and custom build a weapon on the fly. That is a huge, huge misconception. The antigen actually just acts like a key searching for a pre-existing lock. Out of the millions of different lymphocyte clones already chilling in your body, the antigen binds to the tiny, tiny fraction that just so happens to have the matching receptor. So the antigen doesn't create the specificity, it just selects it.
And the way this defense rolls out follows a beautiful, logical flow. First up is recognition, where a rare, perfectly matching lymphocyte bumps into the antigen. That leads right into selection, because the antigen binds to that specific cell. This triggers activation, basically waking the cell up and sounding the alarm. And finally, we hit clonal expansion. This is that magic moment where that single selected cell rapidly divides over and over and over, transforming itself into an overwhelming, highly targeted clone army.
All right, Section 4, the necessity of diversity, preparing for the unknown.
So you're probably asking the obvious question here. How on earth can the body have a pre-existing clone for a virus that literally didn't even exist a year ago? If we aren't building these responses on the fly, how are we ready for completely novel threats? The answer to that puzzle is diversity. Before you're ever infected by anything, your immune system essentially shuffles genetic pieces around in this incredible process known as VDJ recombination. Using just a limited number of genes, this genetic shuffling generates an absolutely astronomical variety of antigen receptors.
This massive library of possibilities is called your lymphocyte repertoire. Your immune system isn't trying to predict the future. It's just preparing for literally everything by creating this vast diversity upfront.
So the core, fundamental mechanism boils down to this. You absolutely cannot have specificity without first generating a massive foundation of diversity. The body builds that incredibly diverse repertoire up front. That repertoire allows for unique specificity in each individual cell, and then finally, that specific fit allows the invading antigen to make its selection.
Let's add some complexity in Section 5, cross-reactivity and nuance beyond one-to-one. Because, biology is rarely super rigid. When we say a lymphocyte recognizes an antigen, it's really easy to picture this single cell wrapping its arms around a giant, whole bacterium. But that is not what happens. The receptor actually only recognizes and binds to a tiny, highly specific structural fragment of that larger molecule. We call this little fragment an epitope, or an antigenic determinant. A single large protein is going to have many different epitopes scattered all across its surface.
And because a single virus or bacterium has all these multiple different epitopes, your immune system doesn't just send in one clone. It launches what we call a polyclonal response. Multiple distinct clones of lymphocytes, each one recognizing a totally different epitope on that exact same invader, get activated all at once. You are literally hitting the pathogen from multiple angles at the exact same time. This structural reality is huge because it means even if a pathogen mutates slightly and manages to change one of its epitopes, the immune system as a whole won't be easily evaded. Those other clones, they're still firmly locked onto their targets.
Now, we also have to talk about cross-reactivity. Since specificity isn't just a rigid, absolute one-to-one lock and key, there's a bit of flexibility built into the system. If two completely different microbes happen to share an epitope that's shaped really similarly, a single receptor might actually recognize both of them. This cross-reactivity can be super helpful, giving you a layer of cross-protection against related bugs. But, and this is a big but, if a microbial epitope looks a little too much like a normal structure on your own healthy tissue, well, this same cross-reactivity can sometimes trigger unwanted autoimmune responses. So ultimately, specificity is really about a receptor's ability to selectively recognize a very particular molecular structure, regardless of where it happens to find it.
Bringing it all home in Section 6, the targeted immune response, the final defense. Looking at this complete journey, it's just amazing. The immune system first relies on this massive, randomized diversity of receptors. Then, when an invader inevitably arrives, specific recognition happens right down at the epitope level. This perfectly leads to the clonal selection of the exact right lymphocyte for the job. That single lymphocyte expands into a massive army, and crucially, all those offspring maintain the exact same specificity as the original cell, which means that the antibodies they secrete or the direct cellular attacks they launch are perfectly, flawlessly targeted solely at the original antigen. The precision of that very first microscopic encounter dictates the precision of your entire final defense. It's just an incredibly elegant, beautifully targeted system. But wrapping all this up actually leaves us with a pretty incredible cliffhanger. We now know exactly how the body flawlessly identifies and attacks a totally novel threat using its vast repertoire and clonal selection. But what happens after the battle is finally won? How does your immune system preserve this incredibly specific experience so that if this exact same antigen tries to return years later, your response is exponentially faster and stronger? I'm going to leave you with that provocative thought about immunologic memory because honestly, that is exactly what you should be wondering right now. Thank you so much for joining me for this explainer, and keep questioning how this incredible biological architecture works to keep you safe.